This is the last lecture for this class. This is the 15th week, and this is chapter 18, or the last chapter we're going to cover. In the week 14, we covered what memory was, and now we're going to look at it from a neural point of view. So we're going to break it down into what biochemicals are actually functioning and creating our memories. There we go. Okay, chapter 18. Research shows that memory formation can take place in the development of simple neuronal changes all the way to complex neural changes. Memory is only formed when neuronal changes take place. Simple neuronal circuits may receive input from more complex superordinate or modulatory circuits. However, some memory requires more complex neural connections, relatively complex networks called cell assemblies. And this is what a cell assembly is. You're doing calculus, and you need all of these connectors in order to understand what you're talking about. These are cell assemblies. So it's not just one neuron, it's a series of neurons, and that is known as a cell assembly. Many of the changes in neuronal structure can be measured physiologically. These changes may be presynaptic, postsynaptic, or both. Many researchers believe that long-term memory requires significant neuronal structural changes. Donald Hebb suggested that if a neuron frequently stimulated another neuron, that it would make a permanent change in the stimulated neuron. And this is where practice does make perfect. And this is one of the reasons why last time I said, how many times does it take to remember something, to get something in your long-term memory? And the answer is the magic number of 3 usually. Now, some people can remember things after one incidence, but a rule of thumb is 3. Maybe it takes 5 for select complex structures, but 3 is a pretty good number. The Hebian synapse is a synapse that has changed because of frequent stimulation. This means that the synapse is not only active in stimulating other neurons in the area, but that it is frequently stimulated as well. Neurons that fire together wire together. This frequent stimulation follows Hebb's dual trace hypothesis. Research actually done by the authors of this text dealing with the weight of rats' brains and a standard environment, an impoverished environment, and an enriched environment proved that rats who were raised in an enriched environment had heavier brains than rats raised in the other 2 environments. The area of the brain that had the most significant growth was the cerebral cortex, but especially the occipital cortex. Further research has proven that much of the change takes place because of dendritic branching. Dendritic morphology has been shown to be constantly changing. Fine filaments extend from the dendrites. These are called filopodia. If this filopodia make contact with an axon, they may form dendritic spines. When a synapse receives repeated strong stimulation, it creates a stable and enduring increase in the effectiveness of the synapse. This is termed long-term potentiation. This can especially be seen if the neuron experiences tetanus, a flurry of intense and constant electrical stimulation that creates a permanent change in the synapse. The hippocampal formation is formed by 2 interlocking C-shaped regions, the hippocampus, dentate gyrus, and subiculum, the hippocampal gyrus. These 3 structures are divided into 3 regions, CA1, CA2, and CA3 from bottom to top. NMDA glutamate receptors acts as an antagonist preventing long-term potentiation. But NMDA does not affect long-term potentiation that has already been established. AMPA glutamate receptors increase long-term potentiation. Activation of NMDA receptors leads to an increase in AMPA receptors. When glutamate is released in a synapse that has both AMPA receptors and NMDA receptors, the AMPA receptors require only moderate stimulation to be activated, whereas the NMDA receptors require a greater amount of stimulation. This occurs because magnesium blocks the NMDA calcium channels effectively, keeping the NMDA receptors from firing. If the AMPA receptors become stimulated enough, it depolarizes the membrane, removing the magnesium and allowing the NMDA to fire. This allows a large amount of calcium to enter the neuron, so the NMDA can only be activated when the cell is depolarized and there is glutamate present. The large influx of calcium activates protein kinases, enzymes that enable phosphate group, the PO4, to attach to a protein molecule. Protein kinase A, PKA, protein kinase C, PKC, calcium calmodulin kinase, calcium MK, and tyrosine kinase TK. Calcium MK affects AMPA in two ways. It phosphorylates AMPA receptors, increasing their conductance of sodium and potassium ions. It promotes the movement of AMPA receptors from the interior of the neuron to the surface of the membrane. CAMK leads to the formation of intermediate-term memory. PKC leads to the formation of long-term memory. PKA leads to long-term potentiation, which in turn helps to establish both long-term and intermediate-term memory. The activated protein kinases not only help to phosphorylize proteins, but they also trigger the synthesis of new proteins. The protein kinases activate CAMP responsive element binding protein, or CREB. CREB in turn binds responsive elements. The elements that CREB binds to are nucleotides. The nucleotides are affected, are in the promoter regions that are affected, are in the promoter regions of DNA adjacent to genes. This changes the rate of transcription of adjacent genes. This in turn alters the growth and differentiation of the neuron. If the protein that recognizes CREB is genetically disrupted, it causes a syndrome that is characterized by intellectual disability. But there are also CREB antagonists. Inducible CAMP early repressor, ICER, produces 4 proteins that block CREB gene transcription. These proteins block CREB by binding to the genes themselves, but do not promote transcription. If too many ICERs are present, CREB will not induce long-term memory. Another CREB antagonist is phosphatase 1, which activates CREB. It is thought that these CREB antagonists exist to pair unwanted memories or to keep them from forming to begin with. The memory process takes place over hours. For the first hour, protein does not have to be synthesized to maintain the memory. But for the memory to form long-term potentiation and hence long-term memory, CREB has to be activated and protein synthesis has to take place. After 3 hours, select proteins that have been synthesized will decrease while others must increase to maintain the long-term potentiation. At this point, the affected neuron will signal the presynaptic neuron to release more transmitter to maintain the signal. This is known as retrograde signal. Memory retrieval requires finding the proper cue to connect the individual with the neuronal bundle that contains the information. When the memory is found, research demonstrates that the structure of the memory becomes unstable and susceptible to disruption from protein synthesis inhibitors. If there are no changes, the memory will be reconsolidated and return to stable status. This is, of course, necessary for memories dealing with a person's behavior where inappropriate conduct must be repaired immediately. Epinephrine affects memory formation by influencing the amygdala. It does this by forcing the release of norepinephrine in the amygdala. If the individual takes propranolol, the amygdala's memory-enhancing effects are blocked. Opioid peptides will also block the release of norepinephrine in the amygdala. Since there are several major emotions, different neurotransmitters and structures come into play with the amygdala to create memory. Adrenergic, opioid, GABAergic, and cholinergic. The amygdala influences organs that are connected to it through neuronal connections, the hippocampus and the caudate nucleus. Propranolol works by taking the emotion out of the memory. The memory still exists, but the details aren't as distinct. The brain as a whole shows a loss of weight beginning at age 30. The frontal cortex loses more weight during this decline than any other portion of the brain. The hippocampus also shrinks with aging, being one of the reasons for elderly to display memory problems. Another area of the brain that degenerates with age is the septal complex that provides input to the hippocampus. Since most of the neurons in the septal complex use acetylcholine as their transmitter, degeneration of the neurons or acetylcholine-producing cells lead to memory problems. Alzheimer's disease is such a memory problem. And that is the end of the last chapter. So we are finished. Okay, well, I'll talk to you later. If anybody needs any help with anything, just give me a call. Drop me an email. I have office hours Monday and Wednesday from 3 to 5, and on Tuesdays and Thursdays between 8 and 10. So nothing on Friday, but if you need any help, just drop me a line at any time, and I will communicate with you at the next possible time. I can turn on my Zoom, and we can have a Zoom meeting at 2 o'clock in the morning if necessary. So talk to you later, and it's been a great semester. I really enjoyed this class, and I'll see you next semester.